Hi everyone. Hi Pandurang. Nisha, good evening. Aditya, a very good evening to you. Meghna, Aditya, Akanksha, and welcome to another session of NEET Bytes. So I guess I have to wait for a few more seconds before your chats load up. And welcome again to all of you to another session of NEET Bytes. And you all know we are discussing molecular basis of inheritance. Right, Nikhil? Good evening. Neha, a very good evening to you. So I welcome all of you to another session of NEET Bytes in Biology by Meritnation, of course. Okay. So I guess everyone is pretty much settled. So I'm waiting for all of us to come up. Anyways, so let us quickly discuss what we are going to talk about in this class. In this class, definitely we are going to talk about our genetic code or the codons. We are going to talk about translation. Then regulation of gene expression is a very important topic over here, followed by HGP and the final and the most innovative technique, that is a DNA fingerprinting. So all of these things will be talked about, right? So I guess everyone is almost ready or pretty much there. Hello, Shriya. Hi, Deepak. And let us all get moving and let us talk about the questions. So I guess everyone is ready and I can come up with the first question of the day. Right, Deepak? Yes. Meghna, let's focus here. Hi, Nita. Okay, so here comes the first question of the day. If there are 999 bases in an RNA that codes for a protein with 333 amino acids and the base at position 901 is deleted, okay. So people are putting up the answers such that the length of the RNA becomes 998 bases. How many codons will be altered? I guess many of you or almost everyone knows the answer here. Hi DJ Bros, welcome to the class. Rishu, good evening. And the answer over here is option C. That is the correct option, right? And I guess all of you know that. So 900 amino acids means divided by 3 will means 3 900 nucleotides divided by 3 will mean 300 amino acids, right? And the 901th is altered. So in total, or is deleted. It's not altered, it has been deleted. So in total, the next 99 nucleotides have been affected. From 98, 9 it became 98. And 99 divided by 3, that means 33. So the next 33 amino acids will be altered. In fact, only 32 will be coded. The 33 will not be coded. The 33 at 1 will not be coded over here because it will lack one nucleotide to be coded, right? So I guess this is almost clear to all of us, and we can move over to the next question. This was pretty simple. Okay. Yes. And the next question is Sai Prasad, good evening. Which of the following RNAs should be most abundant in animal cells? Is it the rRNA? Is it the tRNA? Is it the mRNA? Or is it the miRNA? Which among the four is most abundant among in the and within the animal cell? Okay, Aditya says option A. Yes, Ame, the answer is option A, Nikhil. It is not tRNA. See, rRNA are exclusively present in case of eukaryotic cells, right? Because they are a component of your ribosomes. So they are limited to the eukaryotic cell, and in any point of time, a eukaryotic cell has got at least few thousand rRNAs to synthesize the good number of proteins which are required by the cell, right? And you know how many proteins working as the structural component of the cell, working as enzymes, and in various other forms are present or are released by the cell. So obviously, A lot of RNA is required for that at any point of time, and thus the most abundant form of RNA within the animal cell or a eukaryotic cell is generally rRNA. So that is your answer. Animals can be na. What do you mean to say animals can be na, Nikhil? What is option D? Okay, what is miRNA? Anyone? Good evening, Muhammad Tanveer. Well, just figure it out and let me know, Ame. Right, so you will have your time. And let us move on to the next question, which is also pretty simple. Which one of the following is the starter codon? I guess everyone knows the answer for this. All right. Yes, yes, yes. Everyone knows the answer to this one. There is only one AUG, which acts as a start codon as well as it codes for the amino acid methionine. Right. So it serves the dual purpose. I hope that is pretty much clear to all of us. Right. Yes. And let's move ahead, Akanksha. And let us see the next question of the day. And my next question. Okay, could anyone figure out what is miRNA? Aditya? Okay. Next question is a complex of ribosomes. Arun, Raj, good evening. Welcome to Meritnation NEET Bytes biology classes. Okay, a complex of ribosomes attached to a single strand of RNA is known as a polymer, a polypeptide, Okazaki fragment, or a polysome. And the answer over here, Tanveer, it's option D. Polysome is multiple RNAs, multiple ribosomes associated with a mRNA, right? A mature mRNA which is being transcribed. So you will have a single mature mRNA, and all across it, there are a number of ribosomes, right? So a complex of ribosomes attached to a single strand of RNA. In fact, this is an mRNA to be more precise. Hello. Okay. Nikhil, please focus here. Henry, good evening. Sandhya, very good evening to you. Okay. So, where is Meghna? Jyoti, good evening. Pretty late, huh? Okay. So, I guess this one is almost crystal clear to us. And I can move over to the next question of the day. It's me, the prettiest. Oh my God. I'm Aarti. If you remember me, I came to see your smile, sir. Oh, that's great. Thanks a lot, Aarti, for that appreciation. It will be great if you could have your ID with your own name. Okay. Lakshmi, good evening. A micro RNA is a small non coding RNA molecule found in plants, animals, and some viruses that functions in RNA silencing and post transcriptional regulation of gene expression. This is for Ame. Thank you, Shreya, for putting that thing up for Ame. That's great. Now, Ame, I guess you know what is miRNA. So it is a short form of micro RNA, and Shreya has given us the full detail of it. So, anyway, someone had the time to Google it up. That's great. So, you should thank Shreya for the same. Okay, let's move ahead and let us focus on the next question of the day. This is pretty interesting. So, which of the following is not required for any of the techniques of DNA fingerprinting available till date? Is it zinc finger analysis, which is not required? Or is it restriction enzymes, which are not required? Is it DNA hybridization? Is it polymerase chain reaction? Okay, Tanveer says C. Nikhil says C. What about Meghna? You says A. Okay. Lakshmi says A. Amay, you are right. So the answer over here is option A. What is zinc finger, by the way? A finger made up of zinc. It's a motif, a small secondary structure of a protein in which there is a zinc ion involved. And it forms coordination bonds with this secondary structure motif of the protein. And this forms the zinc finger. So we are not trying to, okay, and we are not trying to basically analyze the zinc finger in a protein molecule when we are talking about DNA fingerprinting. So, therefore, this is not required during DNA fingerprinting, and this is the correct answer. Restriction enzymes are definitely involved in chopping off your DNA sample, right? But before you chop it off, you need to amplify the DNA sample that you have collected with the help of the PCR, right? Thereafter, you're going to apply restriction enzymes to it, chop it off, and then run it on a gel, right? Obtain a gel of it, and then hybridize it with a suitable DNA probe, labeled DNA probe, and view it under the autoradiography using the autoradiography technique, right? So this is pretty simple, standard technique, and this is the figure which has been lifted from your NCRT book precisely, and it is for a purpose. You know pretty well what could be the what is the answer for this. So I hope the figure gave you the clue. Okay, let's move ahead and let us talk about which of the following is required as inducers for the expression of lac operon. So yes, Jyoti, B, C, and D were surely required during your DNA fingerprinting. Here, okay, Arun says the answer is option B, lactose. Okay, I guess everyone is going to say, Nikhil says lactose, Meghna says lactose. What about Dipali says lactose, Nisha says lactose. That's great. I am seeing her answer after a long time. Don't know where she went off. Okay. So the answer is definitely lactose. In lac operon is an inducible operon. Unlike the tryptophan operon, which is a repressible operon, and lactose over here is an inducer. It is not an inhibitor, right? And it binds with the repressor of the lac operon and inactivates it. So we all know this. We have seen that figure or the diagram of the lac operon functioning, right? So I guess let me just pour it out for us. So we all have seen this diagram of the lac operon functioning, right? It is there in our NCRT books. So we all can very clearly see it, and we know this is the repressor which binds to the operator region, right? And this is the inhibitor gene which is expressing the repressor. And this inhibitor gene, when it expresses the repressor is inactivated by the inducer. In this case, the inducer is the lactose itself, right? And thus, your lac operon is able to express itself. Yes, Ame. What happened? Ah, Ame. Well. Please, I guess we have discussed it in the classes, NEET classes. So please go back and watch the recordings or put up your doubt in Ask and Answer forum, right? So, or you can ask it over the Teacher Connect app. So even in the Ask and Answer forum, they will give a reply to you. So since we are discussing a specific topic, not of plant reproduction in flowering plants, we are discussing molecular biology, right now. So therefore, please, I hope you understand. So we'll be talking about this question only. Okay. I'm in 11th and I studied this chapter for extra knowledge. That's great. Priya, probably you are having biotechnology in your syllabus then. Anu, good evening. Okay, let's move on and let us talk about the next question of the day. So, the next question of the day is, yes, Chef Pandey, good afternoon. DNA dependent RNA polymerase catalyzes transcription on one strand, not stand, it is strand of the DNA, which is termed as the template strand, the coding strand, the alpha strand, or the anti strand. Yes, you all know the answer. It is option A. Yes, Payal, you are right. Right, Dipali, that's the answer. So template strand is used to is read by the DNA dependent RNA polymerase, and it transcribes a HN mRNA. Heterogeneous nuclear mRNA is transcribed in case of eukaryotes, right? Heterogeneous nuclear RNA is transcribed in case of new eukaryotes, right? Using the template strand, even in prokaryotes, it is just the mRNA, not the heterogeneous nuclear RNA. What is the difference between the two? I guess you know it. In prokaryotes, there is no post transcriptional modification, whereas in eukaryotes, post transcriptional modifications are required to make it a functional mRNA, right? Anu, just figure that out and let me know. What do you mean by an anti strand? Right, template strand is the one strand which is read by your DNA dependent RNA polymerase, right, and is used to create a RNA molecule. Right, coding strand has the same sequence as the newly synthesized RNA molecule by DNA dependent RNA polymerase, but it contains uracil in place of thymine, and of course ribose sugars and its nucleotides in place of deoxyribose. All right, could anyone figure that out? Okay, I hope this one is done and we can move over to the next question of the day. So, which enzyme or enzymes will be produced in a cell in which there is a nonsense mutation in the LAC-Y gene? So, what do I mean by a nonsense mutation? I must definitely understand that in order to answer this question. I should know the structure of the lac operon, which I have just shown. Arun says it is option C, but that is incorrect, dear. Okay, Meghna says it is option A. Alpha sheet is a atypical, but this is not an alpha sheet. That was an alpha strand, dear. So I didn't know whether they are talking of an alpha sheet or an alpha strand. In any case, so that should not be the secondary structure of a protein. That is a beta sheet we generally talk of. Okay. Shreya, I hope that is clear. You don't use the word alpha strand. Yes, it is an alpha strand, it is not an alpha sheet. So the answer over here is which enzyme will be synthesized if the LAC-Y gene is inactivated by a nonsense mutation. A nonsense mutation is the one which will convert a functional codon into a nonsense codon or into a stop codon. Right? So that is a nonsense mutation. So if in the LAC-Y gene there is a nonsense mutation, the scenario will be this: the LAC-Y gene will become unfunctional, right? And as a result, the LAC-Y gene portion will not be translated, right? This portion will not be translated because there is a stop codon coming up over here, and A will not be also translated. So transcription will be there, but the translation of permease, the translation of transacetylase will not happen. Only the translation of the Z gene will occur. So only beta galactosidase will be synthesized in this case. Right? So I hope everyone is able to understand this reply. And the answer is option A, thus. Shall we move on to the next question? I guess this is pretty much clear to all of us. All right, let's move ahead and let us talk about the next question. And the next question says removal of RNA polymerase 3 from nucleoplasm will affect the synthesis of what? Will it affect the synthesis of tRNA, hnRNA, or mRNA or rRNA? Okay, well, I am talking about RNA polymerase 3, and I am removing it from the nucleoplasm, of course, of a eukaryotic cell, because there is no separate nucleoplasm in a prokaryotic cell. No, the answer is A. It will be tRNA. Remember, RNA polymerase 1 in case of eukaryotes is responsible for the synthesis of rRNA. Right, Akanksha? RNA polymerase 2 in case of eukaryotes is responsible for the synthesis of mRNA. Right, so mRNA is synthesized by RNA polymerase 2. Right, your rRNA is synthesized with the help of, so just a second. So I can say very clearly here, your RNA polymerase 2 synthesizes your mRNA. Yes, your RNA polymerase your RNA polymerase 1 will synthesize RNA polymerase 1 will synthesize your rRNA. Absolutely correct, Akanksha. Right. And that will synthesize your rRNA, and your RNA polymerase 3 will synthesize your tRNA, right? 5 point, I think 5S rRNA is also synthesized by RNA polymerase 3, but RNA polymerase 3 is basically meant for the synthesis of tRNA. For the synthesis of tRNA. I guess that is clear. Yes. Heteronuclear RNA is a mRNA in case of eukaryotic cells, which is synthesized by RNA polymerase 2, right? But it is called as heteronuclear RNA because it has not undergone post-transcriptional modifications. Yes, heterogeneous nuclear RNA. No, it is not heterogeneous nuclear ribonucleoprotein. It is not HNRNP. It is a HNRNA. So HNRNA is the mRNA without any post-transcriptional changes in case of eukaryotic cells. I hope it is clear, Shriya. So don't get confused between HNRNP and HNRNA. All right, I hope this is clear. And the next question is, DNA fingerprinting refers to molecular analysis or profiles of DNA samples. I guess that is the answer, right? Analysis of DNA samples using imprinting devices. So are we analyzing the DNA samples? How much of adenine, thymine, purine to pyrimidine ratio? No, the answer is option A here. Techniques used for molecular analysis of different specimens of DNA. No. Techniques used for identification of fingerprints of individual. Definitely no, Dipali. Not even B. Analysis of DNA samples using imprinting devices. What kind of imprinting devices are we using? We are using various devices like the thermocycler. We are using the gel system, right? To run an agarose gel. We are doing agarose gel electrophoresis. We are using blotting techniques. We are using DNA hybridization as well as thereafter autoradiography, right? So nowhere we are using any sort of imprinting devices as such. Answer is option A over here. Science and Tech by Srivastav Mridul. Thank you for retracting that message. That was okay. I guess that is it for the day. And with this, we are done. Right. And you all know there is a link in the description below, which you can just click on, enter in your details, and enroll yourself for 2020 NEET program. So I hope that serves all of you pretty well, even from a point of quick revision for your board examinations. So we have quickly revised the whole unit, and that is it for the day. So let us say bye bye and see you all again in the next session of NEET Bytes. There will be talking about evolution. So see you all again. Bye, guys. Thank you, Nisha. Thank you, Dhirendra. Bye bye, everyone.